evolved to eat, what is that, vegetables and other things. They are herbivorous, right?
Yeah.
So they have been evolved for that, and of course as Professor Karve said, like, the configuration of the teeth and other things and all. But then we evolved, but then we started to eat whatever was possible after sometimes, right? But then the early man or something like that. I don't know. I'm not saying that, based on this could change over a period of time, as you said. Things could be different. The understanding could be totally different once we have more and more evidence and other things and all, right? But then I don't know.
Yeah. So this is, as I said, see, the flint is a classic example, right? It's a very simple and 3 lakh and 3.3 million years. Is a
Yeah. No, that's what I'm saying.
Yeah.
And the flint also, the Flintstones, the cartoon, that is how the stone is coming from, I think. Yeah. I have one more question, Krishnamurti.
Right.
Your slide, you said, like, one of the timeline was that they started to have agriculture or something like that 6,000 years ago or something like that. You said something. Yeah. And then the beer manufacturing or, beer brewing was 5,800. Within 200 years, we evolved and then started to make beer.
So
no, it's good. I'm not saying it's bad, but then 200, not bad actually.
No, rice was 6,000 years old. That's a field in China. Yeah. So that is what I was impressed that people were
6,000 years and then 5,800 years.
Yeah. So they feel that it could be even older. See, this is based on a clay tablet which is available in Iraq and a beer residue in Iran. But the belief is that if they can document it at that time, probably people were using it little even earlier. So if you go back in 11,000 years ago, the agriculture, the proper cultivation started, irrigation, everything, and maybe in 1,000 years they actually started making beer and wine.
Curious with the numbers, that is all.
Yeah.
Okay, thank you very much.
Yeah.
Just one comment. That drug what is given to soldiers is methamphetamine called Pervitin.
Pervitin, right?
MDA. Yeah. I think we can close here actually, and Professor Madhavrao can.
Thank you very much, Krishnamurti, for a very nice talk. Because to prepare these slides and to take all this information take a lot of time. I know. So you spend a lot of time. Thank you very much for doing this. So now we might.
Professor Rao, can I just intervene a slide?
Sure.
So there was some technical issues with the YouTube live streaming, so we had to stop. I have messaged it in our group also.
Yeah.
So if you are, we have shared the new YouTube link. So if you are sharing with your students and all those things, you can forward that message. Okay. It is in our group, WhatsApp group.
Yeah, thank you very much. So now we go to the next talk by Ashok Giri. Yes.
No. That's Rajiv Kumar.
Who? Next is from who?
Myself, Rajiv.
Rajiv, sorry. I missed it. Okay. I have your idea. Next is Rajiv Kumar. He is going to speak on a very interesting topic. On one side, we are seeing a TV where Deepika Padukone and all those fellows are called, and he is talking about all the. I think very nice to hear. Thank you very much. Come go ahead, Rajiv.
Okay, thank you, Dr. Rao. See, this is a topic which has been in my mind for many decades.
Yeah, very good.
And I have been, this is also my hobby, not the research work, as the Gopi must be knowing very well. Now, I have written around a very big chapter, around 10,000 words and few things very recently submitted, and it is in the press for a book chapter. Now, this is basically, recently it has come. Now, I just want to say, this is the outline of my talk. I'm going to talk about something about the brief history and some sort of about basic, what is the legal status and all those things, and then industrial applications, particularly for the legal cannabis. There will be a difference between the legal cannabis and non-legal cannabis, as per the NDPS and as per also the single drug convention, international. And then I will go for the medicinal applications, research and commercial trends, and you will be surprised to see, and I was surprised. And then, of course, summary and conclusions. And there are some backup slides if there are question answers, more on the technical part, then I can discuss that. Now, coming to the cannabis plant, the minimum time, what it was told is at least 8,000 years before. That is the documented evidence. But people in our Vedas, it is still, perhaps, I don't want to discuss on the dates, but this is what it is documented. In the 6,000 BCE, means 8,000 years. It is one of the fastest growing plants on earth. One of them. Perhaps the bamboo can compete with this. All parts of the plants, like roots, mainly for the medicines, and leaves, as Dr. Karve has been telling, leaves, flowers, seeds. All these things are consumed completely. The treasure trove of phytochemicals, it contains almost 450 to 500 phytochemicals, including cannabinoids, terpenoids, and many compounds. Very rich source of the cellulosic material. It is woody and fibrous and used for making bioplastics and concrete-like materials. The biggest use for this cannabis was also in the paper making. The first paper on earth was produced through the fibers from the cannabis. Cloth also. And many people were knowing that this the ship industry using this cannabis ropes, cannabis cloth for sailing. The fiber is the major crop at that time. Ancient Indian texts like Atharvaveda, Susruta Samhita, and I have downloaded, by the way, just to see myself. And they have considered it as very venerable, very gifted by the gods, and it is also known as Bhang and Vijaya. Now, new uses are being discovered and reclaiming soil fertility. I was amazed to see that Japan is cultivating it in a large way for removing the nuclear toxicity in the soil. It is a remediation also. Now, cannabis has basically three different, the cannabis is a genus from the Cannabaceae, and it has three basic genus. Cannabis sativa, which you must be knowing, it is also popularly known as marijuana or marijuana. And then there is an indica, cannabis indica, which is basically in the Himalayan region. And you can see the difference. This is tall, very tall, around 15 feet, 16 feet, 18 feet high. The sativa and the indica is basically the broad leaves, very bushy, but it grows up to say 6 to 7 feet. And then this is ruderlis, which is basically grown wild, and it is not seasonal. All these things that you can read downwards. However, the basic difference is that these sativa and indica are psychoactive. Ruderlis is almost nothing, near zero, and it is used by the cattle feed and it grows wildly everywhere. In the north, the moment you are even in my fields, on the side where the water is flowing around, it grows, and the people, this cattle feed is used for that. Next one is basically, I just want to make a difference between hemp and marijuana. Hemp is basically where the tetrahydrocannabinol, the basic psychoactive component of this cannabis, is less than 0.3% in USA and 0.5% in India, legally talking. Anything, and then no difference otherwise. And basically, male plant has got that type of ability, and it is harvested for the industrial uses and for the very derived products. We will come back to that. Marijuana is basically a seasonal one and contains much more than the 0.3. Basically, it is around 3 to 4%, 6%, 7% in the palm, but also it can grow up to the content of up to 16, 17, 18% by hybridization and many more technology. It is intoxicating, side effects, but we talk about the THC content and all these drugs, ganja and all this comes from this one. Recently legalized in 27 to 30 states in America for medicinal and recreational purposes. also legalized in India in 2018, by the way. And IIM, our friend already, IIM has, Jammu Kashmir has been given the license, which will come later stage. Now, what is the legal status? Because this is very, very critical to know for most of the people. Well, Indian Hemp Commission was established by the Britishers in 1894-95, and it has got 2,500 pages report. And I have downloaded it. It's easily available on archive.com, and there are commentaries on that one. The basic purpose was that this was the biggest, third largest crop in the country by that time for ages, and it was also consumed by all strata of the societies. So they realized that this is a very good opportunity for the money spinning. Now, as Krishnamurti said, that the Britishers, opium, but it was not only opium, it was basically ganja and charas, which was also considered as a very similar poppy and the cannabis plant. They are basically in the similar region. However, cannabis was basically, they realized that there are two aspects of it. And people in India, it's culturally in every state they are using. But they wanted to have the kill the Indian medical system, which was very, very popular at that time still, because they want to have an allopathy. Now, allopathy has got, as you know, the basis of alcohol as a base. Similarly, in all Ayurvedic preparations and most of the Ayurvedic preparations, the cannabis, the ganja or the bhang are the basic ingredients as the base. And now they wanted to also promote the alcohol. So that is where they thought whether this can be banned or can be taxed. The purpose was they can get money from this one, and that is what the basic aim. Now, in NDPS, which we hear a lot nowadays on the TV, NDPS defines cannabis on the very third article, sub-article. And what A says that what is cannabis is the charas, hashish, that is the separated resin in whatever form, separated from the plant. It is resin which is separated from the cannabis plant. Or the ganja. Ganja is a fruit, flowering or a fruiting part. They have higher content of the THC, around 20 plus. And in that case, and any mixture of there, that is what the total NDPS talks about cannabis. However, in the cannabis definition, the leaves and seeds are excluded. And leaves and seeds are traditionally called bhang or bhanga. And many, banta also, so many colloquial terminology for this one. And that is where it is not banned. In the federal law of the country, it is basically only the charas and ganja are criminalized. They are criminal. You can have, and then there are so many articles that how much you can have. But bhang federally is not banned. It is excisable, just like alcohol. You can have in north 4 or 5 states like Uttar Pradesh, Madhya Pradesh, Rajasthan, now Gujarat, Bihar, Himachal Pradesh, Uttarakhand. They all have the legal shops, government licensed shops for the bhang. And that is where it is so popular in Holi or otherwise also. The community which I come from is a very common. They take it very, very, but it is all leaves which contain very less THC and more medicinal advantages. Now, as I told, CSIR IIM has got the license from a Canadian company in collaboration with. Similarly, now 30 USA, Canada, a few European countries and Netherlands have legalized the recreational purpose also. So it is a recreational purpose as well as medicinal purpose. And of course, industrial purposes anyway. Now I'm talking about the industrial uses, a snapshot. You can see, and this is not what is, it is amazing. This plant has perhaps the only plant which has got so much to offer to the human beings that every part is used. Now, this particular thing, if you can say, the basic thing I will simply put here. You see, it is the superfood is the seed. The In the Himalayan regions, those who are the sadhus and ecstatists, they simply take a, a bowl full of seeds, roast it and take, and they don't need anything else except water. For them, that is good enough food. And they can survive the whole day for that one. It is just like, and it is very much rich in the omega-3, 6, perhaps much better than the quinoa. And next one, you can see it is a food. It also gets the proteinous part also, and flour. People were making atta from this one. Next one is a pepper making. As I told, is the first time, and pepper is still the very common one, which can be done. Bamboo, since it is banned cultivation, that's why it was killed. That was the basic purpose, to kill cotton and cannabis, because the Britishers wanted to kill the economy and take the whole thing, as everyone knows. Next comes the textile. Of course, when the fiber is there, it is a textile. That's what the basic purpose was. And I will discuss in the next this one. It is a comparison with the cotton. Now comes even the molded plastics. Now this is also recent use. Molded plastics, it can be combined with many things. And presently, there is a designer clothes are made because of this hemp. There is a complete showrooms on the hemp fibers in the US. And in India, also in Gujarat, there is a company called Hemp Fibers, Hemp Cloths. Now coming to, you see on the right hand side of your screen, you can see the comparison with the cotton and cannabis. Cannabis hardly needs any pesticides, because no pest can attack it. It is so natural. It has adapted to the nature that it is widely used. It hardly needs any attention. It does not need much fertilizer, because it has got intrinsic capability to kill the bacteria, what Dr. Karve was telling, because of the so much of and the roots are very much capable of killing the bacteria. Now you have to only think the bacteria has to be, and it also grows deep rooted, and that's why it holds water. Now the next one is also, you can see the warmth. It is 300 to 400 times more warmer. That's why in the north regions in China, this is the cloth which is being used even in the Central Asia. And also it has almost 4 times more water absorbing capacity. And that is where it contains. And similarly, the strength of the fiber is 3 times more than the cotton. All these, the only problem is initially it is coarse, but now with the new spinning things, it is made very soft also, and combined with the other fibers. Now this is the general. I'm giving a snapshot of hemp industry applications, which are also modern uses apart from these one, and every part of this plant is being used. Agriculture benefits. It can be soil remediation, soil improvement. Anyway, you simply grow it in the wild and its soil improves. The modern things are many things. I don't want to spend too much time on this one, but purpose of this slide is that it is very much industrially used if it is cultivated freely. Only problem is that it is ganja and other parts also coming from the other piece, so that is being. But now the whole world is going towards legalization, is getting intensified with each passing day in the world. Just last week only, Pakistan has legalized for the industrial applications. India has legalized in 2018 anyway. Now coming to the chemistry, what does it make it psychotropic? There are 3 types of cannabinoids. One is the phytocannabinoid, we can say, that is where the 2 main cannabinoids. You might have heard so much so about the CBD oil nowadays on the TV. So it is basically a tetrahydrocannabinol. The structure is given on the right hand side. And you see the difference between tetrahydrocannabinol and CBD, or cannabidiol. So little. They are functional isomers basically. It's only one is having an etheric linkage and a close ring. The other one is having only an alcohol and a double bond. They all are same, they are isomers, but they have a very different problem. CBD is practically non-psychotropic. It does not give you high at all. THC is highly psychotropic. This is the difference. And it is the potency of any these, the material is the CBD, THC CBD ratio. If they are in the 1.1 to 1.0 ratio, they are best for the medicine. Now, synthetic cannabinoids are also being developed by the actually Israeli scientists have been doing this much better. They have been doing it faster. And the first time this impetus for the research came in 1964 when THC was isolated. And they found that this is the psychotropic material. Since they found that this is the psychotropic material, there must be an endocannabinoid receptor also. So, and then they synthesized very similar to that. You can see the very similar material is called HU210. HU is for the Hebrew University. Endocannabinoids, when there is a cannabinoid receptors, there must be an endocannabinoid. And then in 1992, this same group identified, actually extracted from the brain and called as anandamide. They found that this material is basically similar to the anand of Sanskrit word. That's why anandamide, and its isomer is a virodamine. When amine is there, it's an amide, arachidinoyl ethanolamide. And they also found in the pregnant mammals placenta, and also in the brain, the two arachidinoyl glycerol. No much difference, but they are basically, they are two endocannabinoids which are very popular. Coming to the endocannabinoid receptor system in human body, this is very critical. It's not very technical, but I just like, there are two types of cannabinoid receptors. They are CB1 and CB2. And why it has got so large plethora of advantages of the medicines, and that is what we are going to look into, because it can, CB1 receptor is basically concentrated mainly in the brain part of it, cerebral part of it. That is why the THC acts on the cerebral part much more actively, and that's why you get the brain buzz and many other things. It has got tremendous value. People are used, and there are more than thousands and thousands of papers are coming on this aspect nowadays. One of the highest sought after area of research. And you can see what are the functions it can do, so many functions it can handle. And the CB2 is basically in the peripheral system, immune system, basically. Now, there are papers on the COVID-19 also, because of its immune system. It enhances the immunity like anything, particularly the seeds and leaves enhance the immunity like anything. Now, you can see on the right-hand side how many parts of the body it can affect. So this is where the large number of applications for the medicines you can use. Now, just to give a little bit of why does it, how this they work, I have the neurotransmission, you can read it. But most important part is in the end, which is red font. EC plays a major role in regulating our emotions. I'm talking about the endocannabinoid system, the endocannabinoids, endocannabinoid receptors, which are there. This is the upper side or the right hand side. The first one is the receptor, and then one is the transmitter. And it is slightly different than the normal transmission. That's why I wanted to have it. It is just opposite of the normal transmission of the cell. And those who are interested, we can discuss that later on in the QA. Now, it regulates our emotions, thinking, and behavior. That means actions. They all are chemical based for this one. And the way we feel, that means the way we feel, the way we feel, think, and act. Another very important point, that is the most important part, according to what I feel, reproduction, anandamide and 2-arachidinoyl glycerol was found in the mammalian ovarian system and placenta and the reproductive system almost 100 times more than the brain. And that puzzled the scientists. It was really puzzling. What does it mean? Then it was a lot of research gone into this one, and they realized that it is anandamide / 2-arachidinoyl glycerol facilitate the early pregnancy. Without them, the pregnancy will not happen. The fertilized egg has got the placental connection only triggered by these chemicals. Otherwise, there won't be any attachment, and this basically facilitates the so-called the first communication between the mother and the child is still not developed. And then it also, most important part is that the babies, those who are particularly on the mouse, the clinical work has been done. Same with the other one also. Newborn babies, and they are put the blocker of this CB1, CB2 receptors blocker they are given. They could not suckle the milk from the mother, and that is where they could not get the nourishment. And they found that those the newborn babies were given this antagonist, we call it antagonist, or the receptor blockers, could not suckle. They did not have the ability to suckle the milk, and that's why they died. And this is where this is also very, very critical, that until unless and by the way, colostrum, the first milk of the mother, mammalian mother, has got a very large concentration of 2-H-noyl glycerol and anandamide in it. This is where I think that the people are now debating whether the mammalian development of the mammalians come from this particular cannabinoid receptor systems in the mammals. Now, medical applications. Medical applications of the cannabinoids are basically a large number of things. I don't want to read all these things. You can say that it has an anxiety, means all the psychotropic material, anxiety removal. That's why it gives you the, I can discuss that mechanisms, but that is not, there is no time for it. Particularly, PC controls the hippocampus. That's why it also gets the memory part of it. Similarly, addiction treatment, many people have used this for the addiction removal, cocaine or alcohol. Alcohol is also psychotropic material, by the way, just like neuropathic, and it reduces the pain. It's the best natural painkiller known so far. The neuroprotection, because it is an antioxidant, very high antioxidant. Amazingly, it controls and cures migraine. These are, I'm talking about these clinical trial-based research studies. I have around 100 papers and reviews I have cited in my paper, in this one. It also is a very useful thing is the epilepsy or seizures, because CBD, THC, anticonvulsant, AC modulation of the seizures threshold. So it controls that one. On the other parts, there are so many, like rheumatoid arthritis is very well known, appetite stimulation for people, it is very much known, and there is a drug called Surinabend, and which was basically for the obesity, anti-obesity drug, which was introduced in the Europe because that's a blocker. So they suppress this appetite stimulation blockers, and that's why the appetite was decreased. But then it has also got side effects like suicidal tendencies, because as you all know, the same part of the brain works for the appetite, for the living, as well as for the reproduction. Now, the biggest thing, and now the whole world is after this oncological, because it is known now, it reduces the tumor very well. And apart from this one, after the chemotherapy aspects, the side effects are also reduced considerably, and neuropathic pain, sclerosis pain, it completely, CBD particularly, and THC, particularly CBD is much more important for this peripheral system, gastritis, biological solution problems, reproductive problems, premature ejaculation for many people, infertility, because some people have got the less of anandamide or 2-Ag. This is where the fertility comes in, and that's why this CBD is added, and because that is the same that works on the same receptor. Now, how much time do I have, Gopi?
Yeah, your time is over.
Oh my God! But give me some time, because I think it is.
Because you can closing remarks.
Two, three slides only.
Okay.
Cannabis is a traditional Indian medical system. It has been very well documented, and there are almost 50 different names given in the Ananda Kand, which I have downloaded and got those names. And these names attribute the different applications of the, so at least 44, 45 applications are defined in Ayurveda, our ancient literatures. And they are documented by the Das and Rasso. Rasso is also Ethano Rasso, very well. It is written.
Rajiv, we lost you.
Seems the internet having a problem.
Rajiv, are you there?
Now, I'm just coming to the last few words. This is where the trends I tell you, and you see the okay. So you see the number of papers I have given them in the blocks of the 4 years block. And but you see, after 2012, the legalization started, and there is a significant jump in the post 2012, 15, and now it is 19. So many papers, reviews, books are coming. It is amazing to see. Similarly, in the patents also, I have come. This is the one. We have very comprehensive data on the number of patents on the cannabis or the granted one. It is the same trend. Now, these are the FDA approved commercial cannabinoid based drugs available. And these are the companies, you can say the GW, the UK, and the name is the Sativex or the Nabixolone, and Dronabinol is a basically it's a synthetic drug, as I told you. It's a derivative of HU210. THC is the main component. It is used enzyme-induced anorexia, chemotherapy-induced. This is where in that one, it is FDA has approved it. Migraine is also, you see, the analgesic migraine. Now coming to CBD is also epilepsy-induced seizures. And then so many syndromes. It is basically anti-inflammatory. And on the right-hand side column, you can see who is the owner of that one. Now, this is where the business is. Last slide or my before conclusion, perhaps. The market size of the legal cannabis is a market report of 2020, and you can see the prediction. Today, in today's world, the legal cannabis or legal marijuana market is around 19 billion US dollars. Global. 85% on the right-hand side, you see, is by US. And that is where the trick comes. And they have seen the value of it. Now, it is expected that it will, the minimum, I have got 3 reports, the minimum CAGR, the compounded rate of growth is around 18.9%, which is very high. And you can see it is expected that by 2027, the global market will be 275 billion. And if India, they have also given, if India has what a very large population, India and China, then it will go into 200 to 300 billion dollar business. It's a very big one for a single crop. Now, summary and conclusions. The cannabis, that is bhang or vijaya, a wonder plant, and subhumidity for many, many years. It was demonized only because of the economic reasons by the Britishers and later on by Americans, again by for the same reasons for the alcohol industry. And now I can simply say, the data says that during this COVID-19, when the alcohol in India as well as in the America and Europe, there is an increase of around 300% sale of this marijuana-based eatables, I will simply say, as well as birds and all those things. It is, as the legalization, modern research is paving the way for social acceptance as well as the governmental acceptance for the nutraceuticals and other things. The resulting, the rapidly increasing legalization is giving an impetus for the commercialization, as well as for the patents and publication. India can benefit a great deal in this one because it is a native plant to us. It does not need too much of protection, and it can simply revolutionize the agriculture part if it is combined with the other crops. Thank you very much for your things. I had to rush certain things, but the material was that it was so exciting to me at least.
Thank you, Raju. Yeah, any question? Briefly, you can have 1 or 2 questions. I think there's one from
Rajiv, it's an excellent talk. I have a couple of questions.
Yeah.
See, this as you said, THC is the psychoactive component which is found both in hemp as well as marijuana. But one is the banned item, and other one hemp. You said it's a legal one. So how do you compare both?
See, it is. I think I missed that point, or I could not emphasize that. See, in the hemp is defined as if the THC content in the plant is less than plant product is less than 0.3% in USA, as per the legal definition. In India, it is 5%. Anything above that one is marijuana.
Okay.
Same is called. Okay, thank you.
That is what.
And what about the CBD content? Is it legal CBD? Because it is also present in both this thing.
CBD is non-psychotropic, and there is a company called CBD in Bangalore, in India, and there is a use. There is the TV people are ignorant. There's a CBD oil. CBD oil is also basically is a slightly what is called misrepresented. Some people can tell the THC also into CBD oil. That is why it is a problem. That's illegal.
See, another question. See, it is a mixture of almost 500 compounds. Both. It belongs to the same species. But how do you isolate these compounds? CBD.
Actually, it is so simple to isolate. Any person can do this. First of all, it is an extraction, and then it is separated. The THC and CBD are common. Basically, they need not to separate. There is a pharmacopoeia. There is a whole book on this one, and it is relatively easy to convert CBD and THC also in the laboratory. Very simple condition. I can do in my kitchen, by the way.
Acha, my last question: can you comment on spiritual uses of this hemp?
I did not want to. I did not.
No.
I tell you, in the hemp commission report, it is very well clear that the sadhus and the sannyasis used for spiritual upliftment also, because it can, and the only thing is that I can simply say it is a dose dependency. If you have to intelligently use it, it is a miracle. If you overdose it, but I tell you, the risk is
any overdose is bad.
Only thing is that there is not a single report in the world of the overdosing. I'm talking about the death is the ultimate risk. And ultimate. Now, there is not a single report attributed to the cannabis in the world or marijuana. However, there are in 2017 report from the NIH, the American NIH body, alcohol deaths were 2,272,000 directly attributed to alcohol. Even in India, you can see alcohol kills many more people, cannabis does not.
Thank you very much.
Okay,
Raju.
thank you.
We have to
I have no problem. I can stay till end. If there are some questions by the people, those people can stay and we can discuss a lot.
Thank you.
Okay, thank you.
Yeah, thank you very much. You give the now. Then we have the next talk is from Ashok Giri, who is from National Chemical Laboratory, the chairman of the Biochemical Sciences Division, and he's going to speak on his recent work, where he has published a lot of publications based on his recent work related to the molecules which are isolated and characterized from his group. Ashok, are you ready?
Yes, sir. Yeah, just I'm sharing the screen.
Okay, thank you. Yeah, very good. Yeah.
So, do you see my slides?
Yes.
Yes. Yes.
You can make a full
I can see your lab, your building.
Yeah.
So that means everything is fine. So.
Thank you, Professor Rao, for introducing, and it was very nice to see, nice to hear all three talks, which are very interesting. So I'm going to talk today something what we did in last couple of months, I will say 4 or 5 months. so battling against COVID-19 by repurposing of therapies. So at the beginning, I wanted to say that also the way other speakers said that I am also not working on COVID anytime. So we work on plant-insect interaction, try to understand how plant defend against insects and pests. You have heard my talk in Kashida on Alfonso. What are the secrets of Alfonso? We work on metabolites of Osimum and several other species of Osimum, as well as exotic plants from Western Ghats like Shiropigia, the beautiful fruits or flowers, as well as we work on these anti-nutritional compounds from tomato and potato. So sometime back, one of a PhD student joined in my lab in 2010. His name is Rakesh Joshi, and he brought this computational biology in my group. Now I'm happy to inform you that he is now faculty in biochemical sciences division. Like after PhD, he worked 5-6 years in university, and now he's back as a scientist. So he brought this computational biology work in my group to understand molecular interactions between protein, ligand, and so on. And that we thought when there was a lockdown, there was nothing that we can go and work in the labs. So we thought let's start working on something on the COVID, there is coronavirus. So it's not something that we are working regularly on this. So I don't have to really talk about this problem of COVID-19. It is every day the deaths are changing, but now as per the today's news, we have almost touching 92,000 people are died, and in one day yesterday it's like 1,141 people died. Of course, there is a lot of news also comes on vaccines and drugs. And just to give you a statistic, that there are 36 candidates of these vaccines against COVID are in clinical evaluation. 9 vaccines are in phase 3 clinical trials, whereas more than 146 candidate vaccines in preclinical trials. However, there are no drugs or no therapies available yet to mitigate the COVID-19. So I'm sure all of you know also this remdesivir, which is RNA-dependent RNA polymerase inhibitor, which is highly used, and it's you hear again every day this news, as well as the other molecules like favipiravir, nitrous oxide, as well as ivermectin. So these are the drugs which are used as a repurposing for the COVID-19. Along with several other efforts from CSR NCL that to mitigate or to handle the COVID, we thought we will work on the some of the expertise that we have in group. So what we did is we know that what are the targets of this coronavirus, and fortunately there were 3 papers came in Nature and Science where they described the structure of these targets. These targets I will explain in a minute. And then we thought, since the structures are available, we can do some virtual screening, because we, as I mentioned you, that we work on plants. So we have a library of more than 5,000 compounds. So we also added some from Dr. Mahesh Kulkarni and other groups, molecules, and we try to see the possibilities of using these molecules or structural libraries to screen against the targets of coronavirus, and then optimize the lead and in vitro and in vivo studies. And I'm happy to share with you that the molecules that we identified for repurposing are already in a clinical trial, and that's a very, very good news, by the way. So, how we started this? Basically, what we thought is targeting multiple targets could be a more sustainable strategy. And I think I don't need to mention to this audience that if you target multiple, multiple targets of the virus, it will be actually sustaining for more time. One of the interesting thing is this human angiostatin converting enzyme. This is called AC2, which is actually entry point for the virus. So there is a receptor in the virus, and that's how it enters into the virus. The other is this virus is RNA virus, and all of you know that this RNA-dependent RNA polymerase to replicate. And to process and to maturation, there is something called protease, which is called MPRO, or main protease, which is also required for processing of RDRP as well as other viral proteins. So all these 3 targets were actually very attractive, and not only for us, we selected these three, but of course for others also and earlier also, whenever they were developing these antivirals, these were the potential targets. And since the structure was available from the coronavirus, this COVID-19, so we thought this could be a good option to screen these molecules against this. So just to tell you again, this SA2, like human angiotensin converting enzyme 2, is an entry point, and it presents in the lung, heart, and intestinal tissues. And this is a very promising target for the developing the new drugs. As I said, this is RNA virus. RNA-dependent RNA polymerase is the machinery that amplifies. There are something like 29 proteins in the SARS-CoV-2, and RNA-dependent RNA polymerase plays a crucial role in actually replicating and disease development. So a lot of antivirals were actually developed using this as a target. So of course we use that as a target. And as also mentioned that this protease, which is a papain-like protease, which is very, very important for self-maturations as well as this processing of viral replicase enzyme. It is actually has two catalytic domain. One is chymotrypsin and another is 3C protease like of kind of coronavirus. So very interesting thing is that if you use this as a target, if you see the similarity of this protease with human proteases, there are no similarity. So that's a good target, so that it could be a very specific to the virus and it will not have any adverse effect on the human if you are using this as a target protease. And if you develop a molecule on the basis of this, it will have very less side effects. So we use these three targets, and before that we thought whether this is a good target. So as all of you know, the origin of this COVID-19 is probably from the bats, but then when we looked clustering of these main proteases in different from different viruses, we could see there are 5 clades. Clade 1 is more of a porcine. Clade 2 is avian host, like if you see here, it's like mostly chicken, and the clade 3 is probably from the bat, and that's quite confirmed now it is from the bat. And these proteases actually have very conserved domain. So that means if you develop a molecule against such a nice target, it could have multiple applications. So we have more than 5,000 plus molecule structure library of these molecules, and we added few of them, and we screened something like more than 7,000 molecules. They include several plant metabolites belonging to flavonoids, glucosinolides, anti-tissue compounds, anti-influenza compounds, antiviral compounds, terpenoids, and alkaloids. And several of these phytochemicals were actually showing strong binding to the MPRO, which is a main protease target, as well as the other targets that I was mentioning about. One is this RDRPs as well as a human angiotensin converting enzyme 2 receptor. So we identified several molecules which showed very promising data. But what I would like to show you, because again, all of you know this remdesivir, which is currently used, and it is quite effective. So the molecules that we screen, and these are like very, I mean, as I mentioned you that in earlier slide, we screen more than 7,000 molecules, but I'm showing here data of only 6 molecules, and we identified a dozen of molecules having much more better score than the remdesivir. So just to tell you, that's binding energy. That means like how much complex or how much stable complex it forms with this MPRO, that is main proteases, RDRPs, as well as human angiotensin converting enzyme 2 receptors. And this is a combined score. So what we did is, since this HC2 receptor is the entry point, so we scored it like 60% and 20% to MPRO and 20% to RDRPs. And if you see these scores like CAS, this is combined activity score, we identified that these molecules, firstly 3 molecules, myricetin, for example, delta viniferin and meritylene, these are actually much better than or equivalent to the remdesivir, as well as the antivirals which are known, like for example, ledipavir, dacalastavir, as well as soposavir. They are actually equivalent or even higher than the remdesivir. So we thought that time, like this was somewhere between like end of April or beginning of the May, and we were like found it very interesting. We contacted several people who are involved in the drug discovery, and they help us to really do lot of pharmacokinetics and other activities, and they also felt that this is very, very promising candidates, and it has to be go for further in vitro as well as in vivo and clinical trials. So just to give you the background of these molecules which we shortlisted. So for example, delta viniferin is coming from the black grapes, that is very common. It is known Ayurvedic anti-tissue medicine. It has strong binding with all the 3 targets, and it is already known that it has antiviral activity. So that also confirms what we were saying is perfectly right. These are the molecules coming from the plants. Similarly, this myricetin, which is a glycosylated analog of the myricetin. Again, it is present in Myrica esculenta, which is called Kafal plant. It's a typical plant grown in actually northeast area. And also this molecule was known that it has antiviral activity against HIV as well as other viruses. So that means these molecules are able to really help not only for the virus replication, but to the entry of the virus. So we propose this for Ayush, and we are working on it, developing the formulations from these 3 plants, as I mentioned, grape as well as this Kafal plant. And these, again, as Dr. Rajiv Kumar mentioned, it's not very difficult to extract these molecules because they have very specific properties and they are quite abundant in these specialized plants. Coming back to the known antivirals, so these are the, again, we screen more than 60 known antivirals, and based on these binding scores as well as combined activity score, we've identified 3 molecules which are much better than remdesivir. One is this ledipazavir as well as dacalastravir, and they could target all the 3 targets like main proteases, RDRPs, as well as human angiostensin converting enzyme 2. And again, we come up with these 2 combination. So for example, ledipazavir as well as soposavir and dacalastravir as a soposavir. And this combination we propose for the clinical trial. I'm very happy to tell you that this inputs from the clinicians, particularly Dr. Dhruv Chaudhary from Haryana, he came forward when he saw our paper. He contacted us, and then we developed a proposal together with one of the CRO in Pune, in Pirangut, called Intox. So they will be doing toxicity studies of this combination, as well as this medical institute in Rohtak. With the leadership of Dhruv Chaudhary, they will be doing the clinical trials. So, but what I wanted to give you the message is that these combinations, as far as our calculations are going on, they are much better than or they are going to work very nicely against COVID-19. Another advantage of these molecules is like remdesivir is injectable, whereas both these molecules, or all the 3 molecules what we identified, they can be given as an oral. So that's another advantage to it. So right now the clinical trials are going on. I cannot reveal the whole data, but what I came to know from our collaborator that this trial started last week, and they are saying that they are very encouraging data. But of course, we have to monitor this for at least 30 days, and then we can really go ahead with this. What I'm trying to say that whatever the expertise were available, and we use this time, and I'm so happy that we could successfully show it. The work that whatever we did on this, it's published in one of the journal in USA, and this till this point, like, we have more than 6,000 views, and 17 in last 3 months it has been cited more than 79 times. So that's again the success. Of course, it's a teamwork from CSIR, NCL, Rakesh Joshi and Mahesh Kulkarni were involved, and the students like Shaunak, Sneha, Shiva, Meenakshi and Vaibhav, they actually were screening and calculating from these data. From Intox, Dr. Narendra Deshmukh and Dr. Anita Shog, she is having more than 20-year experience in the drug discovery. She helped us to do the pharmacokinetic studies as well as keeping this combined activity. And based on that only, we proposed to the DBT, which is called BIRAC, and they have funded our project. Of course, PGIMS basically, they will be doing the clinical trial. Dhruv Chaudhary is the lead person, and Dr. Mahesh Gupta as well as Savita Verma, the clinicians will be doing this study. And with this, I would like to have this mantra that Sarve Bhavantu Sukhani, Sarva Santu Niramaya, Sarve Buddhani Pashanta, Sarva Om Shanti Shanti Shanti. Thank you.
Very nice. Very nice, Ashok. So any questions? You will be happy to answer.
Can I ask something, Ashok?
Yeah, please.
Yeah.
Maybe Ashok, you can stop sharing. Yeah.
Ashok, it's very interesting. I just have one, a comment or something I probably need to clarify myself. So when you say about the drug which is working as with respect to the binding energy, it's a completely static overview. But what about the stability of the drug when it binds with the binding pocket of the particular protein, in this case H2 and so on?
Yeah,
so have you checked
very
MD
interesting
simulations or something like that? I mean, in silico.
No, in silico we could not do MD simulations, but this Dr. Anita Chog, she worked on the drug discovery, and these molecules are known molecules. So we have a lot of data available about their stability and the binding. So as per her views is concerned, it's going to be much more effective, and it's very stable complex. So that's what we are actually now.
These are already known molecules.
Exactly. So these molecules are known. Both phytochemicals, they are actually used in the cough syrup and so on. So there is a lot of data available about these molecules, and of course the drugs which are already used for other, like for example hepatitis and other viruses. So it is already, the data is available, and she studied all these combinations, that how we can, what could be the combination, what could happen when these drugs enter in the human body. So
okay, fine.
You mean Ashok?
Yes, sir.
That means you are saying it is repurposing the drug. Is it like that?
That's correct, sir.
Yeah.
Okay. What's up?
Yeah. So Ashok, so very nice. See, I have a question. See, when you want to repurpose a drug, so a lot of things you can avoid because it is already, you have certain sort of approval, pharmacokinetics, another thing is there. But then here, when have you done, before going into clinical trials, have you done the efficacy of these molecules? I know you would have done a docking studies, right? You would have done a docking, right?
No. So we are, so we have not done, but now we propose, and this is what BIRAC is funding for us, that we would like to do the actual studies on the virus.
Yeah, that's what.
that will be done, but DCGI approved for clinical trial. So in parallel, we are doing clinical trial before even.
Okay. So in parallel you're doing, but then you don't know the pharmacokinetics of these molecules towards actually actual viral infection. That is to be done, right?
So
then still they allow you to do clinical trials?
Yeah. So okay. So the molecules that we identified are known antivirals. So the pharmacokinetics and other metabolites.
Yeah. Pharmacokinetics will be known.
Yeah. So that is known. So on the basis of that, since these antivirals are actually currently used, so based on that, they said that you do these studies in parallel, but we will approve. So 3 weeks back, they approved our clinical trials, and our clinical trials have started. So that was the reason that they actually. Because if we succeed, this could be a molecule identified at least from India that this combination works.
Ashok, hello, Ashok, can you hear me?
Yes, sir.
Hello.
Yes, sir. Yes, sir.
No, yeah, my question is, does it have to go through all the 3 stages of clinical trials, like vaccine, or how does it?
Yeah.
Okay.
So, there will be phase 1, phase 2, phase 3 will be there, but then you can jump to clinical trials faster.
That is a bigger jump. Now we are doing with 60 patients, and if we get interesting data, then this will be.
No, but that means infected patients or healthy people?
Infected patients.
Okay, very good.
Yeah. But you have to do both, I think, control and.
Yeah, of course.
All several controls will be there.
Yeah, so several controls will be there. Yes.
Very nice. Thank you very much. Beautiful talk. Any other questions before we close? Very interesting.
Ashok, can I ask a question, Ashok?
Yeah. Go ahead.
Go ahead. Yeah.
So what is the way forward from here? I mean, say DCJ approved this as a fast track and most important, significant, based on the safety profile and repurposing drug. So what is the perspective? What you are expecting from here in 3 months, 6 months, or a year time?
So right now, the companies who have the license to sell this as a drug, they approached us. And they are saying that we wanted to be a knowledge partner to them. Like, since the licensing and everything is done by them, so we can get patent, but we might not get a actually.
So that's my worry, because for repurposing drug, Indian patent law doesn't allow it, right? So my question was actually who will own the IP for an outcome that what you have been striving.
So Mylan is the company. Probably they will be having. But the good part is that we are actually helping them to go ahead and proceed. And they, of course, they approached us based on.
Which is the company? Which is the company?
Mylan.
Pune based. The same thing what they're doing, kit and all that, right? Yeah. My lab is they got Lonavala, right?
No, Mylan.
No, Mylan. Mylan, M-Y-L-A-N.
Okay.
That is a different one, totally.
Okay.
So that is, Jayant, you are very right, because we initially contacted our IP group as well as Urdeep, and we realized that selling as a drug is not possible.
Not possible, yeah.
There is a question from Archana. ACE2 act as receptor for COVID. Does in previous clinical conditions like diabetes, heart disease, do there is a stronger reception by ACE2?
Yeah, I think there are a lot of studies available. Again, I'm not the expert on this thing, but yes, it is known actually.
Okay.
It is known.
Okay. Yeah. Professor Madhura, maybe we can go to.
Yeah, we know next. Very excellent talk, Ashok.
Thank you, sir.
It's nice. You know, we are doing watching the lockdown itself. Thank you very much. Now we have very young, the youngest perhaps among the all.
Young.
Oh,
yeah.
Krishnamurthy is younger.
My heart is really young.
I'm sorry. I stand for. Yeah. So the second youngest. So is Srivatsan from ISER Pune, with whom I know very personal relationship. Very jolly. I mean, not only an excellent academician, and also very, very, very fun-loving man. So Vatsan, please go ahead. He also done a lot of related to COVID about diagnostics. Okay.
Yeah.
He's our treasurer also. So go ahead, Vatsan.
Before Vatsan start, I just want to say that we are stopping the live streaming in YouTube and also the recording as per Vatsan's request.
Recording.